Работает, все слышно меня? Отлично, большое вам спасибо, Дима. Давайте, хорошего дня. Good morning, everyone. Guys, start coming for a lecture. Guys, start coming for a lecture. Today we are going to discuss viruses. Oh, Bhavesh Verma, good morning. You are the very first one. Maybe you will be the only one who come. I hope not. If you find the link, please send it to your division mates so to come to listen. Good morning, everyone. Start coming, guys. Okay. Today's lecture is devoted to the viruses. So you know that. And the second one is coming, Suraj Maurya. Good morning. So, okay, we start now for you too. Then the others may be coming up. So, you heard during the in the previous lecture, you might heard that viruses is not incorporated or included in any classification that we have discussed. Do you remember? We have discussed three domain classification of all living things on the world and five kingdoms classification of everything, all living things in the world. So that's because they are non-cellular organisms. So they are totally different from any other living things. And today we are going to pay attention at least at the most general features of the viruses. And at the very beginning of our lecture, on the very first slide, you see the topic of today's lecture, when we are going to discuss classification, structure, the way of multiplication and of the viruses, and how do we culture them in the laboratory. So, but then at the very first slide, you see also the very first virus that had been discovered. This is see to the left, tobacco mosaic disease virus, and you can see the symptoms produced by this virus, and that was the very first one. And then to the right, you can see the very last one, newly discovered the particular type of coronavirus that had been called SARS-CoV-2, the causative agent of COVID-19 disease. That is the electron microphotograph of this virus with a very bright halo around the virus that due to the very specific surface proteins that gives the virus the appearance of the solar crown. So, okay, we go on with this, and of course, we have to mention the one who discovered the viruses. So it was in 1892, and that was the Russian researcher Dmitry Yosipovich Ivanovsky, who worked here in Crimea, so first in Odessa and then in Crimea in Nikitsky Botanical Garden, and he had been studying the diseases of tobacco plants. And he did a lot of experiments to catch the microbe that he thought was a causative agent of this disease. But then when he mashed the tobacco leaves and passed them through the filter that supposed to stop bacteria, so it appeared that filtrate of these mass or messy is still infectious. So that's why he understood finally that the new pathogen or new infectious agent that he discovered is very small and it can pass through the antibacterial filters. So he used to call it filtrable agent due to the filtrability of the tobacco mosaic pathogen against the bacterial filters. And he also did a lot of different experiments with this, and he proved that this filtrate stay infectious or stayed infectious for a very long while, at least half a year or a year. So, and then he also discovered that this filtrate is infectious because when he poured or watered the non-infectious tobacco plants, then they get infected in a while with this filtrate. And also he understood and he demonstrated that these microbes cannot be cultured on the or in the bacteriological media. They just won't grow there. So later on, in 1899, the Dutch microbiologist Martinus Beijerinck discovered the ability of these filterable agents uh, to reproduce themselves in the medium where he where we have live cells. So he understood the intracellular reproduction of these new microbes. And Beijerinck used to introduce really the term virus because before anything poison had been called viruses, but he using this term, Beijerinck referred to the specific pathogenic molecule incorporated into the cell. So then, these two are considered to be the fathers of virology, a new field in microbiology. So then we mentioned that viruses are totally different from bacteria, and as a group of organisms, they have some common features. So we just mentioned these common features. So then if we would like to give a definition, so the viruses are the infectious agents that do not possess a cellular organization, they are not cells and cannot be seen with the light microscope. So around how about the general characteristics? They do not have a nucleus or nucleoid or whatever and other organelles, but they are particles, infectious particles, that have an internal core, and inside this core, it may have DNA or RNA as a genome, where all properties of these infectious agents are written. So, but then, mm, unlike other organisms, they cannot have DNA and RNA both together. So they may have either DNA or RNA. And usually this genome of the virus is coated with the protein shell called capsid. So some of the viruses may have one extra layer, that is outer lipid bilayer. external to the capsid that is called envelope. So, and we have to introduce a new term. The term is a virion. And what is a virion? This is extracellular, completely done infectious virus particle. So why we underline extracellular? Because as we mentioned, this virus is the virus needs living cells to reproduce. So really in the life cycle of the virus, of the this infectious agent, we may have alternating stages, intracellular and extracellular. So it may replicate intracellular only. So what are the most important properties of the virus multiplication approach? So viruses are obligate intracellular parasites. Obligated means they may multiply only inside the cell because it doesn't have its own organelles, no ribosomes, nothing for proteins production. So it usually take over the host cell biosynthetic machinery to produce the virus-specific proteins and nucleic acids for multiplication. That is called replication. So then, the viruses have a unique method of multiplication that is usually referred to as replication. Why it is called replication? Because as soon as one virus particle enters the host cell, it doesn't divide like other living organisms. By a unique way of multiplication, it produces identical replica of the initial virus particle. So 100,000, even more identical replica. And this replica will be released from the host cell, and this replica will be progeny viruses. So the one virus may uh, inside the cell replicate into hundreds of the progeny viruses. Then next, components of the virion are produced in a different cell compartments. For example, so sometimes in some viruses, for some viruses, the nucleic acids are produced inside the nucleus and the proteins are produced in the cytoplasm. These are the different compartments, right? So then, after the components of the viruses are produced in different compartments, then they come together that we call self-assembly. Uh, and then from the individual components that come up from different compartments, the complete mature virus particles will be produced. So this way of reproduction is called disjunctive way of reproduction. So and then finally infected cells usually die after release of newly formed virions, sooner or later. So if they die if they cell, that gives rise to the new viruses. If the cell dies immediately after the virions are released and they really kill the cell when they are released, those viruses are considered to be cytolytic. This is cell lysis is destruction, right? So cytolytic viruses. But then we may have viruses that may uh, be not so dangerous for the cell, so they may be released within a while by the cell one by one, so the cell won't gonna die immediately, but then you call them non-cytolytic viruses. But then you have to understand that sooner or later the cell gonna die due to disarrangement of the cytoskeleton and the switching of the protein synthesis from cell protein synthesis to virus specific protein synthesis. Then, so this is the basis of the viral pathogenesis in the human body, that they infect cells and cause lysis or disarrangement of these types of the cells. So, classification of the viruses. They can be classified in many ways. So, first, the most simple one is classification of the viruses based on morphology. You can see here that what does it mean morphology? This is size, for example. So here you can see the comparative size of the viruses to compare with the human erythrocyte or with the E. coli bacteria. So then we measure them usually in nanometers, and you can see the smallest uh, uh, make around 24 nanometers, like bacteriophages that are viruses that infect bacteria, or poliovirus that's a human or animal viruses that make the 30 nanometers. And then the largest virus is vaccinia virus that makes around 300 nanometers. And we may have the Ebola virus that is very thin. There's not really very true width of the virus is shown here, or like this. But they may be really very long, up to 1000 nanometers in length. That is the regarding the size of the virus. And here you can see one of the smallest bacteria, chlamydia, that makes 300 nanometers. So the largest virus have almost the same size as the smallest bacteria. Then the viruses can be classified according to the shape. You can see most probably that's how they look under the electron microscope. So mostly they are spherical, they look spherical. Then some of them may look rod shaped, and that's mainly uh, the plant viruses. We don't see this type among the animal or human viruses. Then they can be bullet shaped like rabies virus, or they can be brick shaped like pox viruses, or they can be filamentous like Ebola virus. Also, the very well known pathogen of humans. So that's according to the shape. But then, that is more important, the viruses can be classified based on their structure. Basically, we can distinguish two types of the viruses according to their structure: non-enveloped viruses and enveloped viruses. So inside the virus we have, as you know already, nucleic acid or genome. And this genome that is shown here in non-enveloped viruses is covered by the structure that is called capsid. So it enclosed the nucleic acid, and we sometimes we may call them together nucleocapsid, right? So the capsid is the protein code, and it is made up of repeating identical morphological subunits that are called capsomers here, right? And each capsomer may be composed of one or several proteins. So, and then take a look in the capsid can be can make a monolayer in some viruses, or capsid may be done of several layers of the proteins. For example, in polio or picornaviruses, we have two-layered capsid. And in the viruses like rotaviruses, we may have three-layered capsid, but still it is a capsid, one single structure. Then if you take a look at the enveloped viruses, they also have a capsid with nucleic acid inside. And the capsid may look quite similar with that of the naked viruses. But then the difference is that apart from the capsid, they may have one extra layer that is called envelope or supercapsid. This supercapsid usually is made up of the phospholipid bilayer like this that is derived from the previous host cell membrane and the virus-specific proteins that are usually glycosylated proteins, we call them glycoproteins, supposed to be inserted into this phospholipid bilayer. And they can be polymerized. This can be dimers or trimers or tetramers, those glycoproteins. So they form the structures on the surface of the virus that are called spike-like structures, right? Like spikes. So those spikes are exposed to the external environment and they are also very important for the virus. So we'll discuss it later on. So this compare please the generalized structure of the virions. So we believe that the structure of the enveloped virus is a bit more complex because of this utmost covering. And you can see here that spike projections formed on the surface of the enveloped viruses can be of one type, or that's very common, that we may have spikes of different types. For example, like in influenza virus, we have two types of these spikes made up of two distinct types of proteins. And so finally, we understand that the viral envelope is made up of two portions. One portion that is phospholipid envelope, this thin layer made up of phospholipids, is a cell specific because this membrane is derived from the host cell. That is really the former plasma membrane of the previous host cell. And then the second portion of the envelope is the spike projections, and they are virus specific. So we have host specific cell membrane that now appears as envelope of the virus, and phospholipid bilayer, and virus specific spike projections made up of one or more glycoproteins. Then, if we take a look at the structure of these capsids, then they can be of two different types. You can see here the geometric of the virion, and it depends on the capsomer arrangement in a capsid. You may see here two types of the enveloped viruses, right? And you may see that the inside structure, that is a capsid, can be of two different types. One type is icosahedral and another type is helical. And the naked viruses also can have a different symmetry of the capsid. But then among animal virus, we may have the only icosahedral non-enveloped viruses, where the capsid make a shell and the nucleic acid is laid free inside the capsid. Uh, Then theoretically, the helical non-enveloped viruses exist, but they are not found within the human or animal viruses. They are found within the plant viruses only. So this type, please pay attention, doesn't exist in human or animal viruses. So altogether, we may have four different types of the virus, structural types: non-enveloped icosahedral, enveloped icosahedral, because this is a type of the capsid symmetry inside the enveloped viruses. Then in plant viruses, but not in human viruses, we may have helical non-enveloped viruses, and then the have a lot of the um, non-enveloped helical viruses with the helical type of the capsid in human or animal viruses. These are four basic structural types of the viruses. And here you can see that those viruses that have icosahedral or cubical symmetry, so have capsomeres which are arranged in triangles, and these triangles form a symmetric figure like icosahedron within the approximate outline of a sphere. So that's like adenoviruses. We see them under the electron microscope. We see them as spheres. So, and also quite often, uh, the enveloped viruses also appear as spheres. So, but then apart from the envelope, you can see here that is made up of phospholipid bilayer and the glycoprotein spikes on the surface. Here we have two types of the spikes on the surface. So beneath the phospholipid bilayer, they have a layer made up of so-called matrix protein that mediate the interaction between the internal structure, internal capsid, and the surface envelope of the virus. So, and that is very important portion of the virus. And then inside the virion, we may see the so-called ribonucleoprotein that covers the um, nucleic acid. And if nucleic acid is RNA, so we call the structure ribonucleocapsid, and the protein that covers the nucleic acid, ribonucleoprotein. So if we compare, I would like you to come back to that. No, not to that. If we compare these viruses, enveloped viruses, and the naked virus, so if we ask ourselves a question, which viruses are more sensitive in the environment? Everyone answers non-enveloped viruses because they have one layer, protective layer, because capsid is for protection, of course. But then it appears that enveloped viruses are much more sensitive in the environment for the drying or desiccation, or for the organic solvents, or for the temperature, or proteases. Why? Because this layer that covers them, phospholipid bilayer, is much more sensitive for all those factors, including high acidity of the stomach, for example, and the gastric enzymes. These viruses are much more sensitive for these factors to compare with the naked viruses. Why? Because the protein shell is much more resistant to the factors we mentioned than the phospholipid bilayer. If in the enveloped virus the phospholipid bilayer is destroyed, so these glycoproteins that are on the surface, they are very important for binding of the virus with the cell and entering into the cell. If they are destroyed and this layer is destroyed, so this virus will fail to find the specific receptor on the surface of the cell and will fail to enter this cell. While the non-enveloped or naked viruses bind with the cell receptor with the help of the capsid proteins. That is the difference in the function of the capsid proteins in naked virus and enveloped virus. In a naked virus, the capsid proteins have receptor function, and in enveloped viruses the capsid proteins don't have receptor function because they are hidden inside the virion. They have only the protective function, while the receptor function to bind with the host cell receptor is attributed to the surface glycoproteins like this. I hope you understand these basic things. Once again, we can see the very good picture of the so-called helical symmetry of the internal capsid. You can see uh, how it is made up of. The green one here are capsomeres that arrange in a hollow core coil, and inside this core we have a nucleic acid. Here it is shown pink, right? So then it appears that the capsomeres are tightly fixed with the uh, nucleic acid. So this type of and this is a picture or computer model of the helical symmetry capsid, and here is the um, electron microphotograph. You see the white, very small structure that are capsid proteins that make a hollow core, and the dark line inside. That is the nucleic acid inside the helical capsid. Okay, this we have discussed already, right? Oh, sorry. So then that's few words about, again, a few words about the function of the capsid proteins. So you see, is this a naked virion? So the capsid proteins are responsible for attachment to the cell surface membrane protein. So see, and that's how the first step of interaction between virion and the cell happens. The virion should specifically bind with the cell receptor. That determines the organ specificity of the virus and species specificity of the virus, because the capsid of the naked virus should match with the membrane protein on the surface of the particular cells. Apart from the receptor function, in naked viruses, the capsid proteins are important antigens, because can you imagine if the antibodies will be produced against the protein that is responsible for the binding with the cell surface receptor? Then, and these antibodies will cover the virion, so the virion will be not able to bind with the cell surface receptor. So this function of antibodies are called neutralization or neutralizing antibodies. So if they are produced, the virion is blocked and cannot bind with the host cell and cannot cause infection. And usually this function is attributed to naked to the capsid in naked viruses, but then this function is attributed to the surface glycoproteins in enveloped viruses. They have the receptor function. They determine the organ specificity because they bind with the cell surface receptor with the help of surface glycoproteins, but not with the help of the capsids. And also the surface antigens, glycoproteins, serve as principal antigens of the enveloped viruses. So then, we go on with the classification of the viruses. We have discussed how they are classified according to the morphological features, how they are classified according to the structural features, and now we take a look at how they can be classified based on the genome structure. I would like to repeat that the genome can be DNA, then the RNA are not present, or RNA, then the DNA is not present. And this genome supposed to be packaged inside the capsid. So, and the nucleic acids and the virions can be designed in different ways. They can be single or double-stranded. They can be linear or circular, and they may be made up of one piece or many pieces. So that's usually typical that the RNA can be either single, may have single molecule of RNA, or several pieces, or fragmented, segmented RNA. That's also typical. Segmented RNA is typical for influenza virus, for example. Then the design of the single-stranded RNA also can be different. It can be a positive polarity or negative polarity. That is important. What do we call a positive polarity viral genome? It is defined as an RNA with the same base sequences as mRNA. Such viruses like polioviruses can use the RNA genome as mRNA. And my dears, the cellular ribosomes will recognize the viral positive polarity genome as mRNA. So in these viruses, the translation may start immediately as soon as viral genome in the form of mRNA binds with the ribosomes, and that is direct translation, I would say. Then RNA can be of negative polarity, and this is the base sequence that is complementary to mRNA, but not same as mRNA. So then you can see the simplified structure of the genome of one of the simplest viruses, that is poliovirus. And then it is a good model to explain that in the viral genome we may have two types of genes: non-structural genes and structural genes. What are non-structural genes? These are the genes that code for proteins that are not structural. What does it mean? That those proteins cannot be found in the virion. And also, there will be structural genes that code for structural proteins that make the virion. My dears, take a look here. So here you can see from VP1 to VP4, viral protein, 1, 2, 3, 4. These are the proteins, genes that code for structural proteins. When these proteins are produced, they form the capsid with nucleic acid inside. So that structural proteins are those that can be found in the extracellular infectious virus particle, that is virion. So, but then when the replication happens inside the host cell, these non-structural genes also will be expressed. It means the non-structural proteins will be produced on the ribosomes, but the function of these non-structural proteins is difficult to overestimate. Why? Because there are usually some enzymes and other very important proteins that help in virus replication in the host cell. But then, if the virus replication is over and the virion supposed to be assembled, there's those proteins that are called non-structural proteins will not be enclosed into the capsid. So we won't find these proteins inside the extracellular virus particle. These proteins will be left inside the host cell and not incorporated into the virion. That is also very important. And here you can see the viral genome of minus sense RNA virus. Here we can see that the specific enzymes, that is large proteins, usually also will be produced as structural proteins and incorporated into the capsid inside. And you can see here many structural proteins, and this is the Ebola virus genome where the structural proteins are written, right? Okay. Now, regarding the DNA or RNA genome, viruses can be classified into DNA viruses and RNA viruses, irrespectively of how this DNA or RNA is arranged, right? So, but then we have around 6 families of DNA viruses with, you can see the examples, and around 13 different families of RNA viruses. So it appears that RNA genome is much more popular among the viral families to compare with the DNA genome. So, but then regarding the nucleic acids, in the Western world, there's a very popular so-called Baltimore virus classification. So, and it the thing is, the animal and human viruses can be subdivided according to the Baltimore classification into 7 groups. And among those groups, the DNA viruses are placed in a group 1 and 2. Then RNA viruses are divided into 3 groups, 3, 4, and 5. But then we have extra 2 groups of viruses that may perform so-called reverse transcription of the genome of RT-viruses. So what is the basis for this classification? Not only whether they are DNA or RNA viruses. The basis is how viruses in each group synthesize their mRNA. And it appears that in each group, the so-called processing of the genetic material is different. What does it mean, processing of the genetic material? I would like to remind you that genetic dogma for all eukaryotic organisms is what? So the genome is made up of DNA, right? And DNA should be transcribed into mRNA, and then that is called transcription, and then mRNA should be sent to the ribosomes, and there the translation happens when the proteins will be produced using messenger RNA uh, as a translator from the genetic code to the protein code. That's why we call it translation, right? Or interpreting. So, but then there's a genetic dogma how the genetic material is processed in all living things. So then, in the viruses, we may have much more variants to compare with the eukaryotic organisms. So, because the transcription and translation may happen a bit differently. So then, this is called the strategies of genome replication. So the strategies, so the distinct mRNA transcription strategies represent the hallmark of each virus group. So, and then due to this, Baltimore classification enables us to classify all animal and human viruses to the extent that the genome replication strategy is precisely predictable. Now let's take a look at the groups according to the Baltimore virus classification. We can have 7 groups. So first two groups are DNA viruses, double-stranded and single-stranded. Then 3 next groups are for uh, RNA viruses. Group 3 is for double-stranded RNA viruses with negative and positive polarity. Then group 4 is for positive sense RNA viruses that where the RNA can be recognized as mRNA. And group 5 is for negative polarity RNAs. Apart from these DNA and RNA viruses, we have 2 more groups. One group is for RNA virus like HIV that may perform reverse transcription, and DNA virus like hepatitis B virus that also may perform reverse transcription. Then, irrespectively of how these viruses produce mRNA, then finally mRNA will be sent to the ribosome to produce proteins for translation. That is the basic uh, understanding of the expression of the genome. The protein should be produced out of mRNA. So then, but in all viruses, the way how they get this mRNA is different. So the strategy is different in different groups. So let's take a look at these uh, groups. So group A, group 1, right? That is double-stranded DNA. It has a very close, the strategy very close to the eukaryotic organisms, right? Because if we have double-stranded DNA, so they regularly, it will be transcribed, the minor sense will be transcribed into mRNA, and then this mRNA will be sent for translation to produce infectious the proteins, right? So then, group 2, single-stranded DNA. So usually what is the strategy? Single-stranded DNA should be copied to make double-stranded DNA, then transcription into mRNA, and then protein translation. See, there's a group 1 and group 2 with the DNA. Then group 3, double-stranded RNA. So for these, yeah, I would like to tell that what enzymes may perform the transcription of the DNA. My dears, this is a regular enzymes, DNA-dependent RNA polymerase. These are regular transcriptases that can be found in the human cell also, right? This is normal for the human cell to have these enzymes that may synthesize DNA and also RNA from DNA. So that's why sometimes the viruses from the first and second group may use cellular transcriptases, cellular enzymes, to make the virus specific mRNA. Do you understand what I mean? Okay, then now we come up to the second large group of RNA viruses. See? So RNA double-stranded, RNA positive sense, and RNA viruses with negative sense RNA. Then how they make the mRNA. For double-stranded RNA viruses, like for example, rotaviruses, these viruses should have, group 3, sorry. So these viruses should have their own enzyme inside the virion that is called viral polymerase or replicase Y. Because in the human cell or animal cell, the process of RNA transcription into mRNA never happens. So it means that these viruses from the group 3 cannot find this enzyme in the human cell. That's why they should keep this enzyme physically in their nucleocapsid inside the virion. Otherwise, they could not process their genetic material. So, with the help of viral polymerase or replicase, double-stranded RNA viruses will produce mRNA, and then this mRNA will be sent for ribosomes for translation. That is the strategy. Double-stranded RNA, then mRNA, then protein. So then the group 4 viruses with positive sense RNA. As we stated, these viruses have the genome that can be recognized as mRNA. See, there is no synthesis here. See, there's a dashes here. So it means the group 4 RNA can be recognized as mRNA. So it directly goes mRNA for the ribosomes to produce proteins. And finally, the group 5 single-stranded RNA negative polarity virus. So this is a viral genome. And again, so as in double-stranded RNAs, they cannot find the specific polymerase, the RNA-dependent RNA polymerase enzyme in the human cell because it never existed in the human cell. So they need to keep this viral replicase or RNA-dependent RNA polymerase as a structural protein inside the virion. So then the first step will be transcription from minus sense RNA into positive sense mRNA, and then mRNA will be sent for translation to proteins. So that is the strategy of genome replication in the or genome expression in the viral viruses with single-stranded RNA. Okay, so these are groups 3, 4, and 5. And finally, we should have groups, or we have just only one, group 6, and that is here. See, group 6, positive sense RNA, but this RNA is not identical to mRNA. So this RNA cannot be recognized as mRNA, and the example is HIV viruses. What they need to do to express their genetic material. they give a very special enzyme that is called reverse transcriptase. And this enzyme performs so-called reverse transcription from RNA to DNA and then to double-stranded DNA. Can you imagine? So that is the name of this enzyme is RNA-dependent DNA polymerase. When the double-stranded DNA is produced, it is a double-stranded DNA copy of viral genome. Then from this, the viral enzyme, the reverse transcriptase, also has activity to produce mRNA based on DNA. So it has also DNA-dependent RNA activity transcription. So then when positive sense RNA is produced, the proteins will be produced. That is a unique strategy of all so-called retroviruses that were named uh, because of the reverse transcription. But then it appears that one of the DNA virus, that is double-stranded DNA, partially double-stranded hepatitis B virus, also may have the very sophisticated way of transcription. So it appears that this virus may multiply through the RNA intermediate genome. So to express its genetic material, they may perform reverse transcription. So from first normal transcription from DNA to RNA, and then from RNA to DNA, and then to mRNA. So then they will express their genetic material. This is one of the very sophisticated strategy of genome replication. So altogether 7 groups, my dears. And here you can see an examples of the viruses from every group, right? So then later on you take a look at these examples and we go ahead with the viral replication. So as we stated, viruses depend on the synthetic machinery of the host cell because they don't have organelles. So, and as a result of entry of one single virus into the cell, hundreds of the progeny viruses will be produced. So, and viruses can cause alteration of the cell morphology, accompanied with the degradation or dearrangement of the cell functions. And this process culminates in the lysis and death of the cell. That is called cytopathic effect. Cytopathic effect of the virus upon the infected cell can be not that dramatic, like lysis. Some other changes may occur for a while, but then we stated already that the cell gonna die anyway. So viral replication can be generally divided into several stages. These are the virus recognition of the host cell receptor and adsorption on the surface of the cell, then penetration into the cell, then uncoating when the viral genome will be released. Then this the fourth stage of biosynthesis of viral components. And I would like to underline that in this biosynthesis, we have to distinguish biosynthesis of viral proteins and biosynthesis of viral genomes, because these are two basic components of the virion. So the proteins are needed for the capsid and supercapsid development, right? And the nucleic acid synthesis is responsible for genomes synthesis that's supposed to be enclosed into the viral capsids. So then the next stage is called maturation and assembly, when the new virus particles will be assembled from the proteins and nucleic acids. And then the last stage is release of the viruses or the virions, newly synthesized viruses or virions outside the host cell into the environment. So these are 6 stages of the process, right? But then you have to understand, but to simplify the process, we may divide it into 6 stages, but then every stage is may be performed differently in different types of the viruses. And each virus group or each genus group have unique properties or unique features of almost every stage in virus replication. So we cannot learn them all those features in all together right now, right? Because right now we just discussed the general features of the replication. But then later on, when we will study special virology next year, next term, so then, step by step, we will learn the most medically important families of the viruses. For example, herpes viruses, retroviruses separately. And then we will learn the very specific features of every virus family replication cycle. Okay? So now we in general describe these stages. So first stage, recognition and adsorption. So the infectious virus particle attaches to the host cell receptor. This is a receptor, usually glycoprotein on the surface of the human cell. This is a protein. So, and then we have non-covalent binding of the viral capsid if it is naked virus. So it makes sort of canyon on, for example, rhinoviruses that matches with the cell receptor. And then the barrier attaches to the human cell. Then the enveloped virus usually do it with the help of those spikes incorporated into the envelope. That is example of HIV, that with the help of GP120, that is the surface glycoprotein, attached to the CD4 cell on the surface of the T CD4 lymphocytes, right? These are the best targets for HIV virus. So this interaction is highly specific. Then second stage, penetration of the virus into the cell. So, and then this also may be performed in different ways. So sometimes the whole virion will be incorporated or engulfed into the cell. So usually non-enveloped or some enveloped viruses enters by this way, penetrate host membrane by so-called receptor-mediated endocytosis or viropexia. And then they appear in the cytoplasm inside the endocytic vesicles that are called endosomes. This is the picture, and this is electron microphotograph of the process of endosome development. Some viruses may enter the cell by different way. So, and this is typical for majority of enveloped viruses, because the envelope of the virus is very similar with the plasma membrane of the host cell. This is the host cell and this is a virion. Then when the enveloped virus attaches to the host cell receptor with the help of specific protein spike on the surface of the virus, the process of fusion occurs, and the viral envelope will fuse, see, with the host cell membrane. This way of entry is called fusion. Then the viral nucleocapsid appears inside the cytoplasm. So this is called fusion. This way of penetration is called fusion. Then the process of uncoating happens. It also may happen in different ways, but we at least mention one. So when the whole virion appears in the endosome, then some viruses may fuse the viral uh, envelope with the not cellular membrane but endosomal membrane by acidification of the medium inside the endosome and the uh, proteolytic cleavage of the proteins and the release of the nucleic acid or genomes together with the capsid proteins directly into the cytoplasm. Then, when the proteins are removed from outside the nucleic acids from viral genome, it makes the genome accessible for the transcription and translation machinery. So usually low pH in the cytoplasm, endoplasmic vesicle is lowering of the pH is mediated by the M protein, for example, in the influenza virus. Then we have a stage of biosynthesis, my dears. So I won't discuss it in very details right now, but the sense of the process I hope you understand already. According to the uh, different strategies I used, as we discussed, to perform viral synthesis, and also viral genomes should be replicated. The sense of biosynthesis stage is to produce viral proteins and viral genomes. And then the next stage is maturation and assembly. So the progeny virion should be assembled from the replicated viral genetic material. Here you can see the genetic material and newly synthesized capsid proteins. So, and the action of enzymes and expenditure of the energy for packaging the viral nucleic acid within the capsid proteins are not required. That's why the process is called self-assembly. So the viral capsid proteins will be assembled and enclose nucleic acid, and then in enveloped viruses, it will be enveloped with a cell membrane modified with surface glycoproteins also produced inside the cell, all usually in EPR and glycosylated there. And then we have a stage of release when the viruses will be released from the host cell. The process of release is also different for different type of the viruses. The naked viruses usually can be released by cell lysis. They rupture the cell membrane and release outside. Usually naked virus cause disorganization of the cytoskeleton, alteration of the host cell membrane structure, and the cell lysis. But some viruses, like enveloped viruses, can be released from infected cells by the way that is called budding. So the viral nucleocapsids that we have here in yellow. So N-specific matrix protein leave from inside in the inner membrane of the cell, and then the nucleocapsids come up to the modified cell membrane, and then the cell membrane evaginates and envelope particles burns off from the membrane. So enveloped viruses derive their lipoprotein envelope from the cell membrane or nuclear membrane sometimes. This is called budding, and it usually is not that dramatic, cause not that dramatic damage to the cell. So here you can see the whole way of replication of, for example, coronavirus. Why it is so important to understand the process of replication? Because in the process of replication, we may find the stages of replication that can be used as targets for antiviral treatment. Let's take a look at the cycle of viral replication of coronavirus. So here you can see the most important viral proteins, the spike proteins, the very small envelope protein, the membrane protein, and the nucleocapsid proteins. So the first stage is binding with the cell receptor. Here you can see the angiotensin receptor that is used as the surface protein to which coronavirus binds. So this first of interaction in between virion and the cell receptor can be blocked with some antiviral drugs. So this is anti-entry drugs that may block infection on the very first stage of binding and entry. So this will be the target for anti-coronavirus therapy. Then we have the process of this virus enters by receptor-mediated endocytosis. Then we have a stage of the uncoating. Then we believe that the stage of uncoating also can be blocked. So the release of viral genome out of the endosome. So then the protein that mediate the uncoating the viral proteins may also serve as targets for antiviral therapy. Then next stage, when the viral RNA will be expressed and the viral proteins will be produced. But very often, the viral proteins are produced as not functional proteins, but very large polyprotein. This polyprotein, to make functional proteins, to be converted to the functional protein, should be cleaved by viral proteases into pieces. Then the pieces of this polyprotein serve as functional proteins. Then that makes possible one more way of antiviral treatment. So the viral polymerase activity can be inhibited. So if the viral polymerase is inhibited, so the functional proteins will not be produced, so the structural proteins will not be formed. There will be no mature virions produced. Okay? Then we have stages of viral genome replication. So the viral genome replication also can be blocked by inhibitors of viral polymerases. And you can see here the names also. So it means the process of viral genome production can be blocked by anti-RNA polymerases. So this is one more stage in viral replication that can be blocked. So quite often the assembly and maturation cannot be blocked. So, but then the viral release can be blocked. This type of drugs had been produced for influenza virus, and this is drugs that before that stop the viral release. But then they also, to some extent, uh, try to be used against coronavirus. I don't think it is very effective, but then it works for influenza virus, but not for coronavirus. But then this is the stage that block the release of the virus, newly synthesized viruses outside the human cell. So you see here that the different stages of viral replication in the host cell can be blocked by different types of drugs. That's how we um, may develop antiviral drugs. My dears, most probably the antiviral drugs are not antibiotics, my dears. So please don't confuse between antibacterial drugs and antiviral drugs. The principle of antiviral drugs development is different from that of the antibacterial drugs. So now we come up to the process of virus cultivation in the laboratory, how we are going to culture it. So basic three approaches can be used for virus cultivation. We may culture them in animals, we may culture them in embryonated hens eggs or some other eggs, and we may culture them in cells derived from humans, animals, insects, whatever, and maintained alive outside the host organism. So then the simplest one, I mean the chronic, the first one, of course, the growth of the viruses in the animals and different ways of animal inoculation. You can see here, right? Subcutaneous, intra, um, peritoneal, and inoculation into the bloodstream, and intramuscular, or other ways, right? So, but then how we may indicate whether the virus is multiplying in the animal or not. So usually the animal may demonstrate the disease or the symptoms of the disease or some visible lesions, for example, or symptoms of the disease, or we may see it by the death of the animal. But we usually use susceptible animals, those that can be infected with this type of the virus. So then the second way how to culture the animal virus is the embryonated hens eggs inoculation. So we inoculate the virus containing liquid inside the uh, uh, egg. So there are several ways how we can do this. We may place it directly on the chorioallantoic membrane, right? So, or we may inoculate it into the yolk sac, or allantoic inoculation, or amniotic sac inoculation. But then, irrespectively of where we inoculate the virus into, so we should have some methods to indicate whether the virus is multiplying there or not. Because as soon as we grow virus, we need to know first whether we have virus there or not. And then if we know exactly that the virus multiplied there, then we need to perform identification of the virus like we do to bacteria. But then identification could be really time and money consuming. So that's why in between growth and identification, we usually perform a stage of indication of the viruses. So there's not highly specific tests usually or whatever. But then we need to know first whether the virus is there or not. That is called virus detection or indication. Then, irrespectively of whether we how we perform egg inoculation, we need to know whether the virus is multiplying there or not. So, and the method depends on quite often on how we inoculate the egg, so the embryo. So that is the example of how we may indicate the presence of the virus when we inoculate it on chorioallantoic membrane. Because the viruses may produce, if they multiply, they may produce the visible lesion, so-called pox. So, under optimal conditions, each infectious virus particle will produce one pox. So, we even may use this method as not just quantile but quantitative assay. So, then the photograph reveals the smallpox virus pox formation on the chorioallantoic membrane. But then, if we use other ways of embryo inoculation, we won't see any visible lesions anywhere. Most probably, we need to perform some simple tests to understand whether the virus is multiplying there or not. And the very popular one is so-called ham agglutination. What is it? So if, okay, first again, we irrespectively of on whether the virus is multiplying, the newly synthesized virions will be seen in allantoic fluid. They will be released there. So then, to indicate the presence of the virus, we suck out the allantoic fluid and we think that in this allantoic fluid there is a virus containing the allantoic fluid is virus containing liquid. We may perform several dilutions of this suspension and then we perform the process of ham agglutination of the red blood cells. When the uh, red blood cells will bind with virus, newly synthesized viruses, through the surface glycoprotein that is called ham agglutinin. This is a spike that may bind with red blood cells. So then through viruses, all red blood cells will be agglutinated or clumped together. Then it will be seen in a 96 plastic well plates as a net or umbrella formation. So that will be called ham agglutination. And the presence of ham agglutination will indicate that the virus is in allantoic fluid. If we don't have any reaction there, so then the red blood cells not connected with the through the viral hemagglutinin, so they will just under the gravity drop the bottom of the well one by one, so and to make a pellet, right? So that is no reaction. This reaction is positive reaction and this is a negative reaction. So, and here we have titration. So, for example, the viral containing liquid had been titrated times 2, times 4, times 8, times 16, times um, 32 times 64 and so on, right? So then we may identify or indicate even the amount of the virus there. So, and the amount of the virus will be expressed as the hemagglutination titer. So, what is a titer? Titer is the last dilution where we see positive reaction. So, this is the highest dilution of the virus containing liquid, right? So, 1 by 2, 4, 8, and so on, right? And then the last dilution where we see positive reaction in that one. So that will be the title of the reaction. That's how we describe the concentration of the virus. And in another virus containing liquid titration, we see hemagglutination just in the first one. So it means there's a very few virus particles had been there. And starting from the next dilution, we see negative reaction. So these are almost negative. Then the third way of the viral growth is to grow it in tissues of humans or animals, or sometimes we use uh, uh, insect cells, right? For those viruses that can be transmitted with the vectors like uh, mosquitoes or ticks or whatever. So, out of different tissue cultures, the cell culture is the most common type of culturing. So, we have a lot of cell types. And the cell cultures usually is classified according to the origin of the cells that are taken, the number of generations through which they can be maintained, and through the chromosomal characters. First type is the primary cell cultures. So how do we perform primary cell cultures? Usually the cells can be taken from the mouse, any mammals, embryo, embryonated eggs or so. So we have taken the tissues, then we cut the tissue or explant, then we usually perform centrifugation to get rid of very large pieces. Then the filtrate should be treated with enzymatic digestion. So why? Because the cells should be released outside the stroma, right? But it should be performed in very short period of time. And then we have to wash the cells uh, extracted from the stroma, right? So wash with normal physiological solution to get rid of the trypsin, for example. And then we have to place these cells in the growth medium suitable for the growth of human or animal cells. My dears, this growth or culture media is not used to culture viruses. It is used to culture eukaryotic cells. Usually they may form monolayer like this, or they can be suspended, form suspended culture in the culture medium. So, but then I would like to repeat, there's a media to maintain cells, but not the viruses. But then when we perform this cell culture and we maintain these cells for about 1 week for primary cell cultures, then we may infect these cells with the specimen taken from the patient that we suspect contain the virus particles. And then the virions will infect those cells and develop themselves in those cells because the cells stay alive in our culture media. Then the second type is diploid cell cultures. See, the first one was primary cell cultures, right? So what is the importance of these cells? They cannot multiply. So they may stay alive for a week, but then they die. So, but then a week is enough to infect the cells and to harvest the newly synthesized virus particles within 3 days. 3 days will be more than enough. So then the cells will be discarded. That is a primary cell cultures. No diploid cell cultures. That are cells of single type that retain the original diploid chromosome number and carrier type during serial subcultivations for limited number of times. It means they multiply outside the host. So, and then we may perform about 50 serial passages. So, when they increase in numbers, we subculture, transfer a part of the cells in a new culture medium, and they will grow there. But then finally, after about 50 serial passages, so they will undergo senescence and die because they start to be very old. Usually these are embryonic cells, right? Fibroblasts that have tendency to multiply. So this is how the culture of diploid fibroblasts appear. So, and then the third type is continuous cell cultures. And these, why we call them continuous, they are cancer cells that underwent malignant transformation. So they may grow forever, for many years, and that's really easy to maintain them. And if you don't need them, we may freeze them and store them under minus 70, for example, degrees Celsius. But then, if necessary, we may unfreeze them and they will stay alive again. That's very convenient, but we cannot use these cells for vaccine development because these cell cultures may contain some signaling protein that may cause malignant transformation of the cells. So, so we can see the suspended cells, and normal cells usually grow like this, right? So because they have contact inhibition of the growth. But then the transformed cells, so malignant cells, they usually grow like this because they don't have contact inhibition. So that's why usually we subculture them in a new tube and then in a new tube and then in a new tube. So how can we detect the presence of the virus in the cell cultures? The basic idea is to check the cytopathic effect, because virus growth in the cell cultures frequently produce a characteristic effects, the changing in morphology of the cells. So, and you can see these different types of the cell rounding. So let's take a look. So, for example, uh, that is oncogenic transformation that may occur as a result of Rao sarcoma virus growth. So cell proliferation is the cytopathic effect. Then you can see the formation of the cytoplasmic vacuolization due to the viral infection. That is very typical for many types of the viruses. It is called vacuolization of the cytoplasm. Then the inclusion bodies formation. If we see this section of the brain, so as a result of rabies virus multiplication, we can see the specific Negri bodies formation. That's at low magnification, that is the high magnification. So then, some viruses may cause so-called syncytial inflammation. There's a measles virus or respiratory syncytial virus when many cells fuse together to form multinucleated giant cells. That's also the way how can we identify it. Or as hemabsorption, because the infected cells may absorb on its surface the erythrocytes. Then how it appears at the higher magnification, right? And adsorption. So, and also we may perform some immunofluorescent tests to find the viral proteins or virus infected cells. That how virus infected cells may appear. My dears, I think you bacteriophages is not that difficult. You read it by yourself, but we discussed the most important and difficult issues regarding the topic viruses. With this, I would like to finish. Ian, thank you for coming. At least 7 persons came, but I hope you will take a look at this lecture later on when you will be preparing for the viruses. Goodbye, my dears. Have a nice day and see you next time. Uh, According to the second week, the next lecture will be already progressing to the microbial genetics. You're welcome. Goodbye, my dears.